Hi, Syma. Welcome to another Science at the Times podcast. How are you? Term is ticking away. Christmas is approaching.
Yeah, I think it's exciting. Like, and finally, beginning to see sort of Christmas trees and lights up going up and things, and so beginning to get quite festive. Yeah, I feel quite optimistic and happy.
It's a nice time of year, isn't it?
It is indeed.
And do you think you're likely at any point over the Christmas period to have perhaps one too many glasses of wine and wake up with a headache in the following morning? I mean, that would never happen to you, I imagine, but
No, that would definitely never happen to me, Tim.
so if you've ever had a headache, are you someone who's like, oh my God, I'm gonna die, with a hangover, or do you just get up and get on with it? Where do you lie on that pain spectrum?
I get up and get on with it, I'm afraid. I think I'm just, yeah.
You're tough.
Yeah, I don't know. I don't want to be biased here, but I think it's a bit of a man thing to sort of think you're dying. No?
I've had a hangover. I've had many hangovers when I wished I had died because the pain was so bad. But it's kind of interesting, isn't it? So people, it turns out, like have different levels of pain and ability to experience pain.
Yeah, absolutely. And the bizarre thing is that right now we have no idea of measuring what those differences in pain are and how, what people's sort of levels of experiencing pain are, because of course we only ever experience our own.
But our guests today, they're interesting. It's got two. One's actually a medic, so he kind of even sees people. He goes into the clinic and he's interested in pain and researches that. And the other's a biochemist who kind of understands proteins. And they're going to tell us quite a remarkable story of how they've taken this sort of looking at people who suffer from pain or moan a lot about it, looking at their genes, finding a gene that seems to play a role in chronic pain. So that's like pain that goes on for months on end, and then worked out why that happens and worked out actually the molecules involved.
Yeah, absolutely. Sort of a kind of almost a serendipitous sort of collaboration between two people who seemingly work in quite disparate areas, which has led to the sort of first real understanding of chronic pain at a molecular level. So
And,
shall
and
we?
they've just published quite an important paper on this, haven't they?
Yes, they have. A very high-profile paper in Nature came out a few months ago, I think, and yeah, it's remarkable because it really has linked something tiny, individual molecules, all the way to a genetic link to chronic pain.
Should we get them on?
Yeah, let's do that.
Excellent. Well, it's delight to have two guests on today who are going to tell us about pain and how we might be able to treat it and what causes it. So I wonder if you'd like to introduce yourselves. Simon, do you want to go ahead? You're top of my screen.
Sure, absolutely, Tim. So, hello everyone. My name is Simon Newstead. I'm the Professor of Molecular Biophysics here in the University of Oxford, and my specialty is using high-powered electron microscopes to image the proteins that are present in your cells that move metabolites into and out of your body.
And David.
Hi everyone. So my name is Dave Bennet. I'm a professor of neurology at the University of Oxford, and I have a dual role really. I'm a clinician, so I see patients suffering from nerve injury and pain, but also I have a big research program on the topic as well.
Okay, great, guys. Thank you. So, yeah, we're here to hear about your recent sort of breakthrough work on chronic pain. I think it's probably useful to start. Perhaps, David, if you could tell us about what is chronic pain and what makes it different to other kinds of pain.
Yeah, I think that's a good question. And I guess in simplest terms, you would say that chronic pain is defined by the duration with which someone has suffered from pain. So often operationally, people are defined as having chronic pain if they've had pain that's lasting more than 3 months. But I think there's probably more to it than that. And if I was to explain acute pain or short-lasting pain, that is something that is evolutionarily very helpful to us. It's important, and actually it's very evolutionarily conserved. You go back to very simple organisms, they have a system for detecting tissue injury. And that's because it's protective. If you are putting your body in a place where it might get injured, so you're accidentally putting your hand on, say, a really hot stove, then actually you want a system that can rapidly detect that and cause withdrawal of your arm from the hot stove. And actually, if you it's probably a topic for a separate discussion, but I look after patients that can't feel pain at all. And actually, that is very bad for them because they get lots and lots of injuries in childhood and their teenage years.
So
the point I'm making. Sorry, go on.
Am I right in fact that it's a really rare condition, but people often die quite young from it because they literally can't identify things that are really bad for them that are causing
True.
acute pain?
Yeah. No, that's absolutely true. It's very rare, but it probably only affects about 1 in a million people. And it's hard to explain to people that do feel pain. But imagine whatever you do, you don't get. In a way, pain is a really important teaching signal to the body, and you will. I mean, I have stories, for instance, that patients. I do have one patient, and he shared the story, which is that. He learned that there was a financial incentive if you left teeth under your pillow, that the fairy would come and leave some money.
Oh, no.
You can imagine what's going to happen next. And he literally went to the shed, got his dad's pliers, and pulled a lot of his teeth out for the financial incentive for that. Or you have patients, as toddlers, anyone that's kind of either has a toddler or has had a toddler knows that there are usually points of discussion between parent and toddler. And it can be a very effective way of getting your parents' attention if you just break one of your finger bones, which again, if you don't have that very powerful teaching kind of signal, these patients accrue a lot of injuries, unfortunately. And you're right, actually, the mortality is higher. In fact, the kind of the first patient that was described by Jeff Woods in Cambridge to have loss of function mutations in an ion channel called NAV 1.7, causing insensitivity to pain. That patient actually died before the paper was published because he was kind of acting as a kind of circus performer, and he jumped off a high height and died. So yes, it really shows you that acute pain is something that's really important for us.
So is chronic pain an evolutionary mistake?
I think it kind of merges into an evolutionary mistake, and it's quite complicated because I think one of the differences of chronic pain is that the system starts to become sensitized. And a good kind of example, I guess, of that in simple terms would be if you're unlucky enough, you go to, I don't know, the beach in the south of Spain, and you lie out and you're fair-skinned and you get sunburn. This is just giving this by way of an example. You get sunburn. The skin becomes inflamed, and then you kind of go home and you have a shower of a warm temperature that would normally kind of be pleasantly warm. It's actually become very painful. And the point I'm making is the whole state of the system begins to change. There's an abnormal amplification within the system. And to an extent, you can imagine that for a while that might be adaptive. So for instance, if you were I don't know, Neanderthal person and you had a broken arm, there probably is an advantage that for a while, maybe a couple of weeks, you try and protect that arm, you're not using it, and that will help healing. Of course, there then comes a time where that kind of morphs into a kind of persistent pain state. And sometimes with there's been healing, or there's actually very little injury to the body in the first place. And you've got this amplification, and it's not serving any useful purpose at all, and that's maladaptive, absolutely. So I think there's this kind of transition. Acute pain, I think, is important. It's very important for life, and probably to an extent, we do need some sensitizing phenomenon, because that helps us kind of heal a body part and not overuse it when it's been injured. But of course, over time, very chronic pain is virtually always maladaptive. And we also have some conditions. I suppose I've focused a bit on tissue injury, but some of the most complex chronic pain conditions, we don't find any injury either to the nervous system or to the tissue itself. So there's no evidence of arthritis, or there's no evidence of peripheral neuropathy. Patients can get a chronic pain disorder probably as a consequence of this gross amplification that's happening in the absence of an injury. So an example there would be disorders where you have pain in the absence of a kind of tissue or nervous system injury would be things like migraine, irritable bowel syndrome, fibromyalgia. These are all chronic pain conditions where probably this amplification is very important.
And this is probably a very naive question. Is that what makes it so difficult to sort of treat or combat?
Yeah, I think that's partly it is, yes, because I think the whole state of the system has changed, and I mean, I think it's can give you hard evidence for this, but of course, the bigger that change is, and the more long lasting it is, then you're trying to take a circuit back to normality, and the more challenging it is to bring that circuit back to normality. And also, I think persistence is an issue. So. Some of the drugs that we use may work reasonably well for acute pain, but not so well for chronic pain. And you will have heard the whole problem with opioids. So,
Yeah.
Opioids, they've been used for millennia for the treatment of pain, and they're let's face it, they're often pretty good for acute pain states, and that's why in the past to an extent they still are used, say for post-surgical pain. The problem is when you start trying to use them chronically, there's a number of associated issues that become problematic. So,
including addiction.
In a way, the body adapts. The dose you need to get the same relief goes up, and you also have this incredible problem of addiction, as you say, and that there's a kind of something of an overlap between the central circuits mediating pain and the circuits mediating addiction, and so they're actually often very poor. And with quite a few side effects when we try and use them for chronic pain. So again, chronic pain is more challenging to treat.
So Simon, chronic pain kind of occurs when something's gone wrong, or we've basically just ramped up, we've turned up like the volume knob on normal pain. And you've been able to identify a way to sort of turn it down again, is that right?
So in a way, yes, because what we've been able to do is identify a gene that's associated with patients that have chronic pain, and that gene has a mutation in it. That's where the association comes in, and that identified Dave's group and the consortia that Dave works in to identify that gene, which he then came to me to try and understand the function of that gene. So it's created a new target for us to develop or start to think about developing an understanding between the connection between the function of that gene and chronic pain in the patients that have that mutation.
So what does the gene do?
So we worked out that this gene is responsible for transporting a metabolite called polyamine across the neuronal membrane. So the neuronal membrane is barrier.
Just unpack that for our listener who might not know what a neuronal membrane is.
So of course, cells are separated from their environment by a barrier that is constructed of lipid molecules or little fat molecules. In the same way that when you put olive oil in water, it coalesces to create a droplet that separates it from the cooking water. What you have in your body effectively is to separate cells from other cells. You have a little fat layer that surrounds them, and that fat layer is what we call the membrane. And of course, as you might imagine, as your listeners will of course appreciate, that fat layer is very complicated. It involves a whole array of different molecules, different types of fat, but also proteins that are responsible for many of the reactions in our body that enable us to take in nutrients from our diet and convert that into energy and so on. Those proteins that we call those membrane proteins because they're embedded in that little fat layer. And the protein that Dave's team identified is called a membrane protein because it's responsible for moving molecules across that barrier.
And so Simon, okay, so you can identify the gene. From the gene, you work out you get to your protein. There's still a great deal of work to be done, right? Because just because we know the protein, we don't know at this point what it looks like, what its three-dimensional shape is, and actually what it kind of does. So can you guys talk us through the story? Because now, thanks to you, we do know what it looks like.
We do, yeah. But before I go into that detail, though, I think it's worth commenting on that. I mentioned that molecule polyamine. So most of your readers probably don't know what polyamines were. In fact, that it was also an unknown metabolite to my group as well. So these are basically they're molecules that are positively charged. The reason they're called polyamines is that they are molecules with many different amine groups in them. And those amine groups are positively charged, and it creates a molecule that is able to have many effects inside the cell. Because a lot of our molecules are charged, they're negatively charged or positively charged. So if you put in these polyamines, you can cause different proteins to come together and other proteins to repel from one another and change the network of interaction inside the cell. I'm just going to pass over to Dave, because there was work done prior to our study that had already connected polyamines to ion channel function in neurons, and that's relevant to the pain data that we presented in our study.
I'd take up on that. So I think to give you a general principle, I've spoken a lot about amplification in the sensory nervous system. So to just take a step back, humans are born with a system to detect tissue injury that starts with specialized neurons that innervate important parts of the body, such as the skin, and they're designed to detect stimuli that could cause injury, whether it's extremes of heat, extremes of mechanical stimulation like a sharp knife, or whether it's chemicals. These neurons detect that change, and they will, as it were, transform that energy into an electrical signal, which is a kind of digital signal of action potentials that are then transmitted across that neuron. They travel from, I'm giving you an example of the skin, they travel from the skin all the way up to the spinal cord. And then there's a set of rather complex connections with the spinal cord that ultimately send that signal up to distributed regions within the brain. And ultimately, the brain is the seat of consciousness, so that signal will be transformed into a conscious perception of pain. And the reason ion channels are important is because ion channels, they control the movement of charged molecules across membranes, as Simon was explaining, and those ions are charged, and that is the basis of these electrical signals. So they're incredibly important in setting that dial we were talking about earlier, about how easy it is for those neurons to become activated by those stimuli. And so the neurons which detect these stimuli, they have a bit of a long name. They're called nociceptors, but in short, we could say the kind of your pain, the neurons involved in those pain circuits, and they're there to detect tissue injury. They have a whole suite of different ion channels, which both are attuned to detect those stimuli. So some of them respond to mechanical stimuli, some extremes of heat or extremes of cold. Some of them respond to particular chemicals, and then to turn that kind of initial stimulus into that signal, which I've spoken about, the action.
So what you've been able to do, you've really been able to kind of go from tissue, so the skin, in terms of where we've got nerves that end there that allow us to sort of to feel things as well and experience the world, but also pain, right the way through to understanding kind of how pain manifests, how it gets to our brain and how it kind of causes all of this, right the way through kind of through what goes on where nerves join one another and how signals are sent between nerves. Have I got that right? Is that a sort of a simplified description?
Yeah, I think it's a simplified view. So, I mean, in kind of to come to the punchline, what we find is the protein, the gene that we identified, and the protein which is the product of that gene. So this transporter SLC45A4 seems to be really important in setting the set point of the activation of those fibers that transmit that information within those pain circuits. So actually, kind of, I guess the key experiment that we did there is we can remove using in genetically modified mice, we can remove that particular gene, and so the protein is no longer present. And actually, when we do that, we find that those neurons are less likely to fire. So really the excitability of that pain circuit is suppressed. And that kind of fits nicely with the fact that in humans, when we look at big databases, we looked at UK Biobank, and that's just how we found this in the first place. We find that humans with variants in that gene, that impacts on their likelihood of suffering from chronic pain. So we've got a very direct link, both from human genetics, but then actually causally testing the role of this gene and the protein it encodes in pain circuits.
So
and then what Simon's been able to do, working with Simon, we would then be kind of able to understand the mechanism of that at a much more molecular kind of level. Both the fact that it was a polyamine transporter, which neither we nor anyone else knew before kind of Simon did that work solving the structure and looking at the biochemistry, and then thinking about how that actually, the molecular mechanisms, how that impacts on the excitability of the pain circuit.
So just before Simon's going to ask Simon lots of questions about the sort of structure of the protein, this biobank, this is sort of genetic sequences and kind of questionnaires with people and measurements from people. And so some of those questionnaires about or measurements are about how much pain they experience. And there's tens, if not hundreds of thousands of people in this biobank. Is that or their data?
The UK Biobank is an amazing resource. I'd say it's probably one of the most important and influential funding for biomedical science in the UK probably ever, if I'm being honest. So it's not just tens of thousands, it's 500,000 people took part in UK Biobank. So there'll be listeners to this that will have contributed to UK Biobank. And I guess the reason it's been such a great resource is it's combined lots of things in a very kind of clever way. So people will ask questionnaires about their health when they entered and gave permission to be part of UK Biobank, but they also gave blood samples. And, we've got increasingly detailed genetic analysis of those blood samples. They linked it to their medical records, and in fact, quite a lot of linkage to their GP records, and also there's detailed information about brain imaging, about imaging other parts of the body. So this is an incredible resource of information that is available to biomedical sciences. And actually, kind of coming back to pain, you're asking about pain. So I think it's true to say that everyone's pain experience is personal. So sometimes people ask how you measure pain. Well, actually, really, it's still the case. I can't measure your pain, Tim. If I was going to put a kind of hot probe on your skin, I would have to be asking you, what do you feel? Is that painfully hot? And in fact, there's huge variation between people. Even if you take completely healthy people and you try and work out what, say, their pain threshold is, there's massive variation within the population. And the point being that you can't, you really have to ask someone about their pain, and it's still a very personal, and it's part of the complexity, quite subjective kind of phenomenon, because it's self-reported. So the only way you can get an index of pain is via questionnaires. And actually, with colleagues, we spent a long time working with UK Biobank to improve the questionnaires that related to pain, that could give us more precision about where the pain was, how long it had lasted, impact of the pain, what the quality of the pain is. Pain is not a unitary phenomenon. You can probably think about that. There's some aspects of pain of where it is, and you can give some idea about how intense that pain is, but some pain might be like electric shocks, some pain might be burning, some pain, might have a kind of overwhelming emotional element. There's an emotional element to pain. Sometimes if we have very severe pain, we might feel extremely scared and anxious. So we wanted to improve the questionnaires around pain, and UK Biobank helped us do that. So that meant that we could get data on really detailed pain data on almost 200,000 people within UK Biobank. And then we can link that to the genetic makeup of those participants, which allowed us to look in really great detail to try and see what are the genetic associations of chronic pain. And that's how we came up initially with this particular genetic finding in this gene, was working on that data from UK Biobank.
And it is amazing to go from all of those individual records down to protein structure.
Yeah,
and Simon,
it is.
I think you're going to quiz Simon now a little bit on that.
No, I'm not going to quiz Simon at all. But for me, the fascinating aspect, is this idea that, like you said, we go from chronic pain down to the three-dimensional arrangement of atoms, actually, which is what you guys have done. And I guess coming from the atomistic end of things, for me, that's phenomenal. But I think it'd be interesting for our readers just to understand, Simon, the process of determining the structure of a protein. Because on the podcast in the past, we've talked a little bit about, just like you can take an X-ray of your arm when it's broken, for example, or something like that, turns out we can do similarly for proteins. But for membrane proteins, some of these methods are quite tricky, and your group's world leading in this area. So I was just wondering if you could sort of just talk us through a little bit of some of the difficulties and the process you go through.
Absolutely, yeah, sure, Simon. So when Dave emailed me, I mean, it was literally, I think, 2022. I think I got this email roughly in about 3 weeks' time in 2022, just before Christmas. This email popped in, and it did say, we've identified this gene, and the nomenclature is an SLC, and the SLC stands for solute carrier. Now that's because many of the molecules that make up our food, like the proteins, the lipids, the sugars, we would refer to those as solutes. When they dissolve in water, they go into water, and then you go into your body, and then they get absorbed across your intestinal tract into your blood, where they're dissolved, and then they get around the body. So that's why they're called solute carriers. And then the carrier is referring to what I was saying before. The proteins carry these solutes across that lipid, that fatty barrier into cells, and enable the cells' metabolism then to use those to create energy to sustain life. So we've been looking at these proteins for many years now, about 20 years, to really understand how these carriers recognize the different food items that we take into our body, but also how they take different drugs like antibiotics and antivirals. If you take a tablet, that tablet has to get into your body, and they're using those same solute carriers to do that. So the way we determine their structure is we have to isolate them from that very fatty barrier. And that's challenging because the molecules that you use to isolate these proteins, the chemicals that you use, sorry, are exactly the same chemicals, realistically, that you use to clean your pots and pans with. So we call them detergents in the lab, and they are detergents, but they're the same. They're really doing the same thing that your laundry detergent does. You're getting a fatty stain, as you will know, sticks to your clothes a lot more than a non-fatty stain. And the problem is trying to get that fat out of your clothes. So you use a detergent to dissolve the fat. So what we do in the lab is we use a very mild detergent, much milder than Fairy Liquid, I should say. For those of us old enough to remember the tagline to the advert, much more milder than, or more milder than. But they gently extract the proteins from this fatty barrier. And they keep, and the reason why it's gentle is it keeps its three-dimensional state. So I'm sure you've covered in the past, molecules that sustain life have a three-dimensional shape, and that three-dimensional shape gives those molecules the properties that evolution has honed into them to do whatever it is: convert molecule A to molecule B, to digest a protein in your stomach, make sure that your cells in your skin don't come off and stick together, so on and so forth. So we need to understand the three-dimensional structure of those molecules in the same way that, a blueprint of a building is a two-dimensional image. It tells you where the walls are, where the windows are, where the plug sockets are. But unless you see the building in three-dimensional space, it's really hard to understand the implications of having the sockets at one end of the room or the windows facing the sunlight and so on. So hopefully, that gives you an indication of how important it is to see molecules in three dimensions. Life is 3 dimensions, molecules are. So how do we do this? Well, for many years, we used a technique called X-ray crystallography. X-ray crystallography was very powerful, but it required us to have proteins that formed crystals that we would then expose to X-rays, and the X-rays would create a pattern that we would then be able to interpret and determine the three-dimensional structure. For this project, we actually used a new technique that has been rapidly evolving over the past 10 years, which is called cryo-electron microscopy. So there, what we're doing is we're using electrons in place of light. So the microscope is the same setup, really, as a light microscope, except rather than light, we're using electrons, and we're accelerating those electrons to under a voltage, and then firing them at our particular protein that is in the sample stage. Everyone remembers probably a microscope from school. You have a sample stage that's illuminated, and then you have an objective lens where you can look at the object. So in an electron microscope, we take our proteins, we put them on the sample stage, and we have to freeze them. So we put them under incredibly cold conditions, and that holds them in place and protects them from the electrons that are then going to be used to create an image of that particle onto a detector that sits beneath the sample stage. And we essentially then generate a set of images from each of those proteins that are the molecules that are in the ice, and that we can use to then reconvert those images into a three-dimensional structure of the molecule we're imaging.
Fantastic. And then from that structure essentially is the organization of all the atoms, right? And so, yeah, I mean, for me, sort of having, I don't know, grown up I suppose as a chemist before the advent of sort of high resolution cryo, and this is fascinating to think that, yeah, we've gone from organization of atoms all the way to kind of understanding pain. But so I guess sort of moving away a little bit from the structure of the proteins, the concept of chronic pain, does this move us forward in being able to combat chronic pain?
I think it definitely moves us forward in terms of understanding, and that's partly because there've been some hints that the molecules transported by this transporter polyamines may have a role in pain, but really it wasn't a kind of field which had been studied in a lot of depth, and this gene has never before been linked to pain. So I think it's really helpful to find these kind of, I guess this whole idea of, say, starting with human genetics is it can open up completely new frontiers of research. And I would say that this gives us a kind of completely new handle on the mechanisms that may underlie chronic pain. And actually the fact that we've been able to link that directly to the way the pain circuits function, I think is very helpful. So I think, number one, it gives us new understanding of chronic pain. But it also gives us a molecular target that we may use to treat chronic pain. And I don't want to get ahead of myself, so I think you've got to be realistic. It takes a long time.
How long until the drug's there then? I mean, I know that.
I'll give you an example.
Are we talking a decade? Are we talking?
I'll give you an example. Hold on, I'll give you an example which shows my age, which was, I remember in, I think it was 1995, that John Wood published a Nature paper showing he had discovered the first TTX resistant sodium channels. That's a particular subtype of sodium channel that we now call NAB 1.8, which was selectively expressed in sensory neurons. So those neurons that we were talking about earlier that detect tissue injury. And he published that in 1995. The FDA approved a drug called suzetrigine, which targets that very specific ion channel. So it targets it specifically. It doesn't have a blunderbuss approach of silencing all vodka sodium channels. It just goes for that one. And that was approved in America last year. So wow. Okay, you've almost got it. It shows you the lead in time between basic fundamental understanding. I mean, we all knew. I remember with that paper coming out, we all knew in the field this was a major discovery because we all thought that there were going to be sodium channels that were specific to sensory neurons, and that there'd been predictive data of it, but we had no idea what the molecular identity is. And then the length of time it took to both study that in more detail, but in particular develop drugs that could selectively silence that sodium channel and no others. And that was a real challenge for chemistry, because I don't want to divert too much into sodium channels, but there's a lot of similarity between them. So trying to specifically silence one and leave the others untouched was a chemical challenge. And then to do the clinical trials to show efficacy, that just shows you realistically. I mean, the thing is, the 10-year is a kind of thing. It's easy to trip out. Everyone kind of says they make a scientific discovery, it's going to impact in 10 years. I don't know is the answer, but all I do know is we need we're not going to make any progress if we don't have the fundamental foundational knowledge. And what I'm saying is this kind of foundational knowledge of linking this gene very specifically with pain. And I should say, actually, now people are basically putting these biobanks across the world together. And now that's been done with over 1 million people's genomes. And actually the association is just getting stronger. I mean, that makes sense in terms of human genetics. The bigger the sample size you have, the more power you have. But the human genetic linkage between pain and this gene has just got stronger. But this is foundational knowledge. It definitely is a something that we would like to think about therapeutically targeting, and Simon and I are already kind of making plans along those lines. But that's how long it takes to transform that into a new drug.
Good luck. I hope it all goes swimmingly. And when when we're older and grayer and the drug comes out, perhaps we'll be able to get you back on the podcast. But we better wrap up because we're running out of time. But thank you both so much. Really fascinating work. Lovely to see the kind of medicine and the chemistry come together to make some major breakthroughs about, as you say, something that's causes people real difficulties in their life, chronic pain. So thanks for joining us. Thanks for coming along, and good luck with the next steps.
Yeah,
thank you.
Syma.
Guys.
Okay, well, Simon, I thought that was really fascinating, but I'm probably going to be in my 80s by the time they've got a chronic pain cure. But I mean, it's possible it could arrive, right?
Yeah, I think so. And that sort of rate at sort of going from identifying a target all the way to a drug is on current. It's a current trajectory, but that may well speed up in future, right? Especially with AI and other methods. So, and they've made the first breakthrough. So hopefully things will happen a lot faster in future.
And I love their enthusiasm for it. Sometimes it was a bit hard to get a word in edgeways, but you just love that when people are talking and they're so excited about their research. It's just fantastic.
Yeah, I mean, because Simon's my colleague here in biochemistry, I've heard him talk about protein structure many times, and it's as you said, that bubbling enthusiasm is fascinating, and I think for our listeners and viewers, it's kind of in science, it's what keeps us going because, as you said, so the drug will come many years down the line, but the individual steps on the pathway are so exciting.
And so, I mean, it's an important bit of work because lots of people, as I said, suffer from chronic pain. These opiates, they can treat it, but they're not very good. People get addicted, they get used to them. So this does offer some sort of hope. Now, as you'll know, one of our past guests was awarded a Nobel Prize for chemistry shortly after we had him on. If a drug comes out from this, I mean, this could be something where another major reward goes to some of our podcast listeners. You never know.
I know, absolutely. I think we're getting pretty good at spotting now.
Come on our podcast and win a major prize. Is that what you're saying?
And listen and watch our podcast to find out who is going to win the next big prize.
No, good idea. But no, I thought that was really enjoyable and really promising. I'm sure people who kind of suffer from pain, things like arthritis, rheumatism, or just sports injuries that take ages to heal. It's kind of...
Yeah, and I think what was fascinating was David's comment that sometimes we don't know why. So it's not, can't attribute it to rheumatoid, arthritis or anything like that. It's just pain and you can't find the cause.
And it's not just inflammation. I always thought pain was just about inflammation, but clearly not. There's this other stuff. So, yeah, it's all in your head or all in your genes.
That's right.
Anyway, I better stop waffling. And I'll see you in a week or two's time for our next guest, who I'm hoping is going to be someone who's written a book on human extinction and when it might happen.
Well, let's not be too doom and gloom, on that happy note.
I think it's a little way off. We should be able to get to the end of the podcast at least.
Excellent.
Okay. Right, have a good one, Syma.
Bye.